Well, hello everyone. My name is Shalyn McCoy, and on behalf of the foundation, the Atlantic Salmon Conservation Foundation, I'd like to say welcome to today's webinar. I always like to say that the series is possible because of part of Government of Canada that is one of our partners. So for today's webinar, we're really pleased to present Dr. Kurt Samways. He is originally from Regina, Saskatchewan. Dr. Samways moved to Atlantic Canada after obtaining his Bachelor of Science and Master of Science from the University of Regina. He earned his PhD at UNB, and he's specializing in freshwater ecology and was appointed as a researcher associate in the Department of Biological Sciences in St. John. He is also part of the Canadian River Institute, which is hosted by UNB and dedicated to making every river a healthy river. He's worked extensively in applied ecology to restore river ecosystems and recovery fish populations, particularly Atlantic salmon in Canada and internationally. He is the lead research scientist of the Fundy Salmon Recovery Program for endangered inner Bay of Fundy Atlantic salmon, studying the effects of captive reared adults on fish fitness and ecosystem health. The project, also a Parks Canada collaborative initiative, has become a foundational element of this research chair program. So with over a decade of food, web, and fish-related studies, Dr. Samways will be continuing his research on a larger scale, bridging salmon recovery and ecological restoration efforts currently underway across 5 Atlantic national parks. So I'm very pleased today to present you Dr. Samway, and he will be speaking on the studying salmon along in natural gradient recovery as Parks Canada Research Chair. So welcome today. And of course, we're going to allow at the end of the presentation 15 minutes for question and answers, which I will help monitor, but we'll make sure that I want everybody to know that that possibility will be at the end of the presentation. So Kurt, welcome, and I'm going to pass it on to you to do your presentation.
Excellent. Well, thank you very much, Shalyn, for that introduction, and very much thanks to the Atlantic Salmon Conservation Foundation for inviting me to speak today. It's a real honor to be able to speak about this unique research chair position that was established by Parks Canada, and about the multi-park salmon project that I am very fortunate to be a part of. So I just want to start off by showing you a map of, obviously this is Atlantic Canada, and these are all the national parks that are throughout Atlantic Canada. And in particular, I want to highlight these 5 parks. So there's Fundy National Park and Puget National Park in Brunswick, Cape Breton Highlands National Park in Nova Scotia, and then Grossmore and Terra National Parks. And probably to no surprise to many of you that all of these parks have Atlantic salmon within their boundaries, and all have existing Atlantic salmon conservation and monitoring programs. And so what's really unique, I think, about this situation, this setup of these 5 parks, is that the rivers within these parks span a relatively large geographic area. And so, but even with that large geographic area, they all are characterized by similar conditions capable of supporting both juvenile and adult Atlantic salmon, and therefore it provides a unique opportunity to study the common drivers of population decline and or recovery, as well as changes in the variability and ecological conditions of these salmon's natal rivers. So here's another map of Atlantic Canada, and showing the national parks, but also the designatable units, or the designatable salmon units, salmon stocks that are found within each of these parks, and as well as showing these different stocks, showing their current conservation status. So as you can see right off the bat, there's a gradient of population status across these parks. So if we look down in where Fundy National Park is, in the inner Bay of Fundy, that population is critically endangered and is the only population currently listed and protected under the Species at Risk Act. So they are really critically endangered down there. If we move up into where Kujmakwack National Park is, and that population, it's a special concern because that population is declining. Many of us are familiar with the declines in populations in the Miramichi, for example, which is very close to Kujmakwack National Park. Cape Breton Highlands National Park, up there in tip of Nova Scotia, I think is unique because the park actually spans 2 designatable units. So on the western side, there's some concern, whereas on the eastern side of Cape Breton, that population isn't listed as endangered under CSIWIC. And so most of the work that I'm doing is on the eastern side. And then you go up to Newfoundland, and those populations are, they're not considered to be at risk at all. However, when we start looking at river-specific populations, we start to see some things that are a little bit different. So again, here's just some plots of changes in adult abundance in these various study rivers in these various parks. We start at the lower left where the Upper Salmon River is. You can see that in the 70s and 80s, the population was fairly high. So in most of these plots, you can see different colors, or unless you're colorblind like me, you'll see different colors. I do know that there's different colors, I've been told. But in that early time, that population was in the dark green, meaning they were very healthy. And relatively quickly, it dropped off to the early 90s, where that population was virtually extirpated. There was some years with adults coming back, but there was years that had no adults. So that's what led to that designation. If you look beside that at the Clybourne Brook, so that's in the Cape Breton Highlands National Park, again, you can see this population decline from really healthy to very low numbers. And so that's what's leading to these endangered status there. In Kujmakwack National Park, they don't track adults the same way as some of the other parks. It was a bit difficult, but if you look at kind of that regional population, again, you can see the general trend of the number of adults is declining over time. If you look at the top left, that's Trout River. So that's a river in Grossmoor National Park. And so although that regional population is said to be not at risk and healthy, and many rivers within Grossmoor National Park are in fact quite healthy, Trout River, as you can see, is not doing very well at all. So their adult return numbers are very low and getting down to critical numbers. As of 2017, they only had 13 adults returning, which is really leading them to consider what needs to be done. And then you have Terra National Park, and in the north, whoops, what happened there? In the Northwest River, that population declined, and there was a lot of concern, and through various conservation efforts, which I'll get into in a little bit later, that population has actually recovered and now actually has a small retention fishery on it. That retention is set by adult returns and various parameters. But I think it's a great example of where conservation has worked and brought back not only a fishery, but a retention fishery. So, a large proponent of Canadian Atlantic salmon restoration has been through Parks Canada. Of course, Parks Canada has a lot of protected natal spawning rivers, right? So there's a lot of salmon spawning rivers residing within park boundaries. And so as salmon populations, as we have these salmon populations across these parks, we know that there's this gradient of population status or spawner densities, right? So again, we go from this virtually extirpated to fully recovered. And because of this gradient, we're provided a unique opportunity to bring these 5 parks under one study umbrella to start looking at common metrics associated with salmon populations and salmon recovery. Along with salmon, of course, Parks Canada has the mandate to protect, preserve, and restore, and so they monitor a number of parameters within the parks to see how things are changing through time. And so on top of salmon, other common freshwater monitoring measures would be aquatic invertebrates, and water quality, just to name a few. And so again, because we have 5 parks, varying populations of salmon, varying kind of geographic locations, varying ecological conditions, we can start to look at freshwater productivity and ecosystem function associated with salmon, not only at the local scale, which is what we commonly have to work with, but we can start to look at it across a large regional scale. And I think this is very exciting to me in particular. As I'm sure you are all well aware of, salmon restoration can and has taken on many forms. With most of this salmon restoration work occurring at the freshwater level. So there's been efforts to increase salmon returns that have focused on various methods such as stocking of eggs or juvenile fish or adults, as well as focused on habitat recovery, working on laws and policies to regulate and mitigate anthropogenic impacts, as well as a variety of social programs that will help to engage community and community efforts in conservation. Parks Canada is very involved in salmon conservation and employs or has employed and participated in most, if not all, of these various conservation measures at one time or another. And so by looking at and across a variety of these methods, we can actually begin to understand maybe what is the appropriate time and strategy that conservation and restoration should be deployed. We know when it gets really bad, we need to do something, but should something be done a little bit earlier to mitigate those precipitous declines? We don't know. That's something we need to understand. But having these parks, again, I'm going to mention it many times, but it's a unique opportunity, I think, to have all these parks with these different populations and ecological gradients. And so we need to take advantage of this. It's a unique opportunity. I was recently asked a question about, well, why salmon? Why not something else? All parks are doing incredibly valuable salmon conservation and restoration work in their own right. And I think that's unique to the region, right? We have multiple parks in relatively close proximity who are all doing really valuable salmon work. And so the idea came that when we started looking at these programs, not all programs, not all methods, not all monitoring practices, et cetera, were being done in the same manner across the parks. And of course that makes sense. You have different parks, you have different park mandates, different situations, and so these different programs evolve independently. But there's a common theme of salmon, and that's what brought this all together. And so the intent of this research chair that I'm very fortunate to be sitting in is not to take away from what has or is being done in terms of salmon and salmon conservation and ecological monitoring, but to bring all these parks together under that that united theme of salmon to widen the scope of what is being done and what we can learn from these collective efforts. And so through these conversations, this led to the establishment of the first ever Parks Canada Research Chair in aquatic restoration. And this provides an opportunity to step out of our individual rivers that we tended to only work in and look at patterns and processes associated with salmon and ecological restoration on a large multi-population scale. Of course, this same approach can be applied to other issues or other species that are common across multiple parks and other in the region or other parts of the country in the future. But it started with salmon because we have, again, this close proximity and this common interest of such an iconic species. What is a Parks Canada Research Chair? It's not the nice Barca lounger that my dad was hoping I was going to get, that he could use at some point. It is very different. But the idea was to have someone who could develop common metrics that could be applied to all parks to look at salmon and ecosystem changes across different risks, restoration strategies, and across this ecological gradient, right? So I guess the definition is why it's in quotes from one of our documents is the Parks Canada Research Chair in Aquatic Restoration. It's a collaboration for research and recovery of Atlantic salmon using regional knowledge and the best available science to restore ecosystems, prevent population extinction, connect people, and inform the future of salmon conservation. That is really the heart of it. And so this Parks Canada Research Chair was established in 2019. The term is for 5 years. Hopefully it will be renewed. That's my little plug to Parks Canada. It's hosted at the University of New Brunswick. I'm situated on the St. John campus. One of the reasons why UNB was chosen, there's a long-standing history of research being done between the university and Parks Canada, and I don't just mean for salmon, but multiple projects. So there's already that relationship. And UNB has demonstrated to Parks Canada that it's a leader in research and education. You know, it's been working quite closely with Parks Canada for over a decade. UNB is ideally positioned to host a research chair position to focus on and design various studies in aquatic restoration initiatives. This opportunity also provides UNB students with unique hands-on training opportunities, and I'm going to talk about that a little bit later on in the talk, because these opportunities, help better prepare them for a career in conservation biology, but get them doing applied science, which isn't always available to many students. So once this research chair was established, we sat down and said, well, how do we drive some of this research? Where do we go? How do we integrate all the parks? We want to make sure that what is happening, what is being looked at, not only is similar across the parks, but addresses each of the park's individual needs. And so there was a workshop that we had in 2020. It was called The Return of the King. That was the name of the workshop. It happened in February of 2020, so just before the COVID shutdown, which was really beneficial, I guess, for us, because really lucky, because even though some of the activities weren't able to proceed with because of COVID, we had a really strong framework, and we'll talk about that framework in a second, but that framework really allowed us to begin some of the work, but at least plan for the work and have everything laid out for when activities could resume. So this workshop was a large collaborative workshop, and it set the stage for really this multi-park restoration program that we have going forward. And the workshop aligned Indigenous perspectives, restoration knowledge, academic capacity, communication resources, as well as just various partner networks, and brought everyone together to achieve a common goal. And so we did have attendees from a wide range of groups. Of course, lots of Parks Canada staff. So we had people from all 5 national parks, all 5 participating national parks. We had people from Parks Canada National Office. We also had representatives from other parks from across Canada, from BC and to Nova Scotia. So this was seen by Parks Canada as a very important initiative. Of course, we had First Nations partners. Myself and the Fundy Salmon Group worked really closely with Fort Folly First Nation, and so they were, of course, an integral part of that. We had Unamaki First Nation Institute there as well, representing Cape Breton, because there's a lot of collaboration there as well. Of course, we had UNB, not only myself, but other people, other faculty members who have various tools or capacity that could be seen as beneficial to the overall project. And of course, we had other members of various the salmon community there to participate as well. So the purpose of this workshop was to sit down and collectively identify threats, drivers, and actions that are important to salmon conservation, important drivers of either declines or recovery. And so from this, this multi-park project that includes these 5 national parks was able to come up with a collective list of items. So this is a very detailed, messy slide. I don't want you to focus on too much right now, but this workshop, like I said, identified threats. So we have a list of threats. These are common threats to salmon that are found across the 5 national parks. We had a number of common drivers that are driving these threats, and we also came up with a number of action items that could be used to address some of these threats. And so from this, there's all these arrows connecting them all. Again, don't get too caught up in those. But on the right side or in the middle is the kind of the green rectangle. And so that was really the core Atlantic salmon recovery project. And so I just kind of blew it up for everyone to see. But really, there's 3 pillars within this core project. Right, there's Atlantic salmon, of course, and so we need to focus on Atlantic salmon restoration, Atlantic salmon conservation, recovery. But Atlantic salmon don't exist in isolation, and if we only focus on salmon, we may be missing out on key variables, key parameters, key understandings of what is driving salmon, what is helping. How is salmon helping the aquatic ecosystem? So the aquatic ecosystem, as you can see, has many arrows pointing to it. So of course, the aquatic ecosystem, freshwater ecosystems in particular, are a big part of this as well. And of course, there's community stewardship, because we can do all the science and all the research we want, but if we don't get it out into the public, if we can't share it, if people don't know about it, did it really happen? Does it really matter? So that's a really big part of it. So through this workshop, this project began by we developed certain elements of successful recovery methods. And we looked at producing a best practices approach. So each park had contributed their kind of independent salmon restoration actions. And we all looked at all the projects, all the parks looked at it through a kind of a conservation standards framework. And so the final product of this, as I've talked about, is this core salmon recovery project. Is really working at multiple scales simultaneously to provide empirical data that will guide future management decisions on optimal timing of intervention, but also connecting these projects and these networks through common ecological measures of either restoration success, and these will provide actionable items to better increase or enhance the effectiveness of salmon recovery. So we had this workshop, and through this workshop, we established a number of regional objectives. And these regional objectives were based on those identified threats. So we were looking at the threats, and we wanted to know how could we address these. And so actions that were identified through this workshop provided a helping hand to better formulate these objectives to address the threats. Once we had these overarching regional objectives, we established the primary research chair objectives for this project. And so there was three main research chair objectives. So the first was to use an ecosystem-based approach to assess ecological function. So we want to understand how these aquatic ecosystems function with salmon, without salmon, and with some sort of salmon restoration. Next one was develop regional metrics to assess the ecological function that are associated with the 5 multi-park restoration projects. And then the last one was make evidence-based recommendations on the ecosystem impacts resulting from active restoration activities at each park. So to do this, we had to establish some common monitoring measures. And this took some time because there's a lot of directions that this could go, of course. But the idea wasn't to create an entirely new system, but rather work with existing programs, existing monitoring, building upon those, but use existing infrastructure, existing efforts to answer these questions. And so the common metrics that were established were algae or biofilm, that primary productivity. Of course, that's the foundation of food webs, stream food webs, river food webs. Benthic macroinvertebrates, so they are a very common biological indicator of river health. And again, all parks are participating in collecting these benthic macroinvertebrates, part of the cabin program already. Salmon, of course, they are the targeted species for salmon for these restoration initiatives. Again, all the parks are collecting salmon data. And so using these common metrics that are being collected in some way, shape, or form, we can also bring them together in maybe a new fashion to measure ecosystem function. So we want to integrate the various trophic levels into a common metric that represents the trophic diversity and the ecological function of a river. So don't forget, when we looked at that map, we saw that there was a lot of variation in population health or size of the Atlantic salmon population across the parks. Of course, there's that geographic variation, and so all of this variation allows for these common metrics to tell a story of how rivers respond to recovery efforts, and therefore, again, when might restoration actions be most impactful? And I just want to remind you that, again, Parks Canada is doing these metrics already. So my job was just to realign and refocus some of these existing efforts to ensure that they're just collected in the right locations. Again, because we want to link them back to salmon, and in a systematic manner, such that they can be directly comparable across all the parks. So once we have these metrics, we really needed to come up with a plan. And so we needed to take steps. And we have to take steps because we can't just do things all at once. We have to sort of dip our toes in the waters before we know what is the right thing to do. So when you start a project of this size, we need to first understand what's going on. We need that baseline data. And that's that assessment part of it. From there, we can move into restoration. Those are the things we need to do for recovery. And then as we proceed, we get those results back, start to see the whole picture. And then it's really important that these results feed back into an adaptive management approach. So these results are going to show us what we need to do to adapt these various approaches as we move forward. So that first step, that initial assessment step, there's a number of projects that I want to highlight, in particular these first three. So one is being conducted by John Robinson. He's a master's student candidate, and he's looking at how salmon populations, and particularly juvenile salmon populations, have changed through time across the parks and under various conservation methods. So whether it's pre-collapse, various stocking methods, various different restoration methods. Another one is by Lauren Sear. She's another master's student candidate, and she's looking at smolt ages and smolt growth. So of course, smolt are an important measure of population. You quite often use them as different measures of population health. And so looking at how smolt ages and growth has changed, again, through time and through various restoration strategies. And the third one is by Kate Gingles, another master's student candidate, and she's looking at these ecological changes that are so important to salmon conservation, salmon restoration. So how do primary productivity, how do ecosystem function change across this gradient and across various restoration initiatives? So we have this baseline of assessment that's going on. The next part, of course, is restoration. And so restoration takes various forms. One is the Fundy salmon recovery model. So this is that kind of smolt to adult supplementation model where smolt are collected from the river, from the Parks Canada rivers, Upper Salmon and Point Wolf rivers in Fundy National Park. They're taken to the world's first marine conservation farm. It's located in Dark Harbor on the island of Grand Manan. Those smolts stay there from 1 to 2 years, depending on maturity. Once they're mature, they're released back into the wild to spawn on their own, and that's a big part of this is natural spawning. Once they're back in the river, they are on their own for natural mate selection and all the environmental pressures. They spawn, they deposit their eggs. We're finding that there's increased egg deposition with these fish. What's key is that when the eggs hatch and those juveniles are in the river, they are free of captive exposure. So the rivers in Fundy National Park are the only rivers in the entire Inner Bay right now that have exclusively wild hatched juveniles. And so that's a big part of this program, is producing the most wild-like fish possible. So Fundy's employed this strategy of smolt to adult supplementation. Cape Breton Highlands National Park is also doing a similar project, similar program. Although instead of rearing in the marine setting, they're rearing at the aquatron at Dalhousie University in a freshwater rearing facility. But it's still the same idea: help those smolt get past that mortality of that early stage, get them to maturity, and put them back in to be on their own. Kujavik National Park, they have a different approach. So to try and bolster their salmon populations, they're planting eggs. So they use these Scotty egg incubation baskets. So they collect some adult salmon returning to their rivers. They're spawned. Those eggs are placed into the incubation trays, and these are placed in the river. They hatch, and those juveniles are then spend their time in the river before they leave as smolt and hopefully come back as adults. So you can see here, there's various degrees of success, but over a couple of years, they've really honed their craft, and they're getting a very high egg survival, which is very, very encouraging. Again, there's not a one-size-fits-all model, and so I mentioned earlier how Terra went from really low returns in 2001 to having a small retention fishery, over 2,500 returns annually. This was done through community engagement. They suspected poaching being a big problem, and so they worked with the local community, local town, local watershed groups, and through a lot of hard work, but building really strong relationships, they were able to turn this river around where there's a retention fishery. I mentioned earlier when we looked at the individual rivers on that map that Trout River and Gros Morne is really low numbers, even though all the other rivers in the park seem to be doing quite well. Again, they suspect poaching to be a big issue there, and so they're starting a working group similar to that of Terra Nova to take that community-based approach to their conservation. So if you think back now, we've established some of these the assessment objectives and studies for this multi-park project. There's various restoration actions that are being deployed. And so now what's really exciting, what I'm always excited about, is looking at some of the early results. So here's some of the population assessment results by John Robinson. So this is looking at juvenile densities across the parks. So these densities are in numbers of fish per meter squared. And so you can see, not surprisingly, in many of the parks, many of the rivers, the population declining. There's some variability in some of the estimates. Of course, that's due to a number of things that I will let John, if you have any questions about that, I will let John answer those questions. I don't want to mislead anyone in why some of these, the variation, but a lot of it has to do with how the data, electrofishing surveys were conducted and things like that. But overall, populations have declined. We're starting to see increases in Fundy National Park in recent years. We still have a fair ways to go. So here we have Fundy in the top left, Cape Breton Highlands top right, Gros Morne bottom left, and Cluj-Quack on the bottom right. Terra Nova's electrofishing surveys, their program is a bit different, so they don't do the same river each year, but they have a number of rivers that are done in different years. But again, you know, we still have a lot of very important juvenile density information that can inform us of what's happening in the park. It's great to have these densities calculated and plotted to see what's happening. But when we start looking at things in a really close, or start making some direct comparisons, that's when we're starting to learn about some of the really interesting things. So one of those things is when we looked at Fundy National Park compared to Trout River and Gros Morne. So here, this area, you can see where those juvenile densities fell to virtually zero when that population collapsed. But through the years, their salmon restoration initiatives, we're starting to see these numbers climb back up to where they were, you know, before that population totally collapsed. So there's a lot of positive results happening there. What's interesting, and maybe not in a positive way, is again, here's where Fundy was at that collapse, and here's where Trout River is now. So although other rivers in the park are doing good, Trout River is not doing well, and it's really where Fundy National Park was back at that time of collapse. And so they're at that point of where some sort of restoration needs to happen, right? They're at that critical point. The road to restoration in Fundy has been long and hard, and so seeing these similar trends of decline in other parks can really tip us off to help avoid those catastrophic, you know, collapses. And it's this regional scope that allows us to do this. And without the research chair and the scope of this project, we may have missed identifying, you know, a critical point like this. And last thing I want to say about this is, I say restoration action is critical, and it is. Restoration doesn't necessarily mean stocking. Restoration takes many, many forms, and we have to apply the right tool to the right situation. Another project that I want to talk about is looking at smolt ages and growth. And again, smolt age and growth is that common metric used to assess health and status across salmon populations. So if we look at the left-hand side first at smolt age, what we are seeing is that when by releasing adults and allowing natural biological processes, natural mate selection, natural spawning, the juveniles experiencing the river free of human intervention, we see that the age at which smolts migrate is getting older, right? And so we see this increase in age. And so there's an inversion to the age structure to older smolts, and this makes sense, right? It would be detrimental to put a child into the real world before they're ready, and many restoration or stocking initiatives when we're stocking juveniles it's kind of like that. We're putting them out before they're ready. This trend of older smolts is what is observed in other reference rivers within wild populations within the Bay of Fundy, within the inner Bay of Fundy. And so it demonstrates that the method that of restoration that Fundy is deploying is producing fish that are more wild-like, right? And you know, if we compare these traditional stocking methods, so highlighted here early on when they're releasing fry and par, you see much younger smolts, a lot of one-year-old smolts. And when you allow that natural spawning to occur, we see this transition to more older smolts. This is a subset of the population. Lauren is currently completing the entire scale library. So by the end of her master's, she'll have all of the individuals from Fundy, from the Upper Salmon River, aged, as well as she's expanding this to other parks as well. The other aspect of this is the growth rates. And the first thing we notice is that we see this trend of increasing smolt ages as we did before. This is just another way of looking at it. But we also see that there is 2 periods of increased growth rates, right? So I've highlighted 2 periods here. And these time periods correspond to adult releases. So there was a pilot project started in 2010, which led to the current Fundy Salmon Recovery Project. So that pilot project was done by Corey Clark of Fundy National Park. And so that led to the current project. But what this is showing, this is actually showing that the ecosystem function of the river is being affected by adult salmon and the adult salmon that have been released back into the river. So increased function is being observed not only with more fish, more juvenile fish, as John was showing in his research, but in their biology as well, in their growth. And so we're starting to see the ecological connections as well. I can't talk about salmon, I can't talk about rivers without talking about marine derived nutrients and ecosystem productivity. I think anyone who knows me knows that's kind of my jam. This is hot off the press from Kate. She's just starting to look at these results. So these are some stable isotopes results from biofilm that were collected in both kind of treatments, so with salmon and reference without salmon sections of the river, across different time periods, across different parks. And so I don't want to get too much into it other than we're already starting to see differences in the patterns of these marine-derived nutrients, of ecosystem productivity, with similar patterns across some parks and differences across other parks. And so it's I'm really excited to see what all comes out of this, but this is brand new. The other part of her work is not just looking at that biofilm or that primary productivity, but I mentioned earlier kind of all the trophic levels. And again, fish, the invertebrates, the productivity, these things are already common metrics in the parks. She's just bringing them together under one analysis. So individually, if we look at just salmon or just the invertebrates, they might tell a certain story of things are okay. But it's not until we bring them all together and see how they're functioning collectively where we get the complete picture. And so this might be what the food web looks like, without salmon. And once you introduce salmon, you introduce this new food resource, you might see a shift and we might see a real dependence on it that lasts not just the season, but can last throughout the winter, which can be very ecologically important. So stay tuned for her results. They should be coming very shortly, and by very shortly I mean hopefully by the fall. But these are some of the interesting results that we're already getting. And of course, don't forget though, results only matter if we can feed them back into the existing program to learn and to adapt. And so we have a lot of different projects currently underway that are feeding back into the various restoration projects, programs, but also highlighting some new innovations. And one that I want to highlight for you today is a project being done by Josh LeBlanc, and is in partnership and really driven a lot by Fort Folly First Nations, their habitat recovery program, as well as Parks Canada. And so collecting fish from the wild and putting them in captivity, whether it's a marine conservation farm or a hatchery like the Mactapac Biodiversity Facility. When you bring fish into captivity, there's a challenge to keep them alive and how to grow them. And they're looking at previous studies. Survival varies greatly from less than 30% to greater than 90%. Most of these studies are in freshwater hatcheries, and we know that marine survival can be quite variable. So one of the tasks one of the things that Josh has been tasked with is looking at how do we get these smolts to survive. So through standardizing practices of how these fish are collected, how they're handled, how they're transported, and a big part is that transitioning from natural wild feed to a commercial pelleted feed. And so through creating a krill oil, soaking the feed in krill oil, dusting it with krill, minimizing disturbance when he feeds. So you can see on the left, there's Josh looking at those smolt, trying to minimize his disturbance there. Various feeding methods. He was able to greatly increase survival. And so as you can see this year, we had between 80 and 90% survival, which is fantastic from smolt. So these are the numbers of smolt going in the spring, so May, June, to as of December, when the last count was done. That's where we're at. So he's been able to look at these various practices and employ them to greatly increase survival. So the next part is, well, okay, great, we have them, we can make them survive, but how do we grow them better, right? What is the best way to feed them to produce a wild-like adult? We fed fish virtually the same way for over 100 years. That 2 to 3 square meals a day. It grows them, it keeps them alive, but it's not what wild animals encounter. So we want to look at how can we introduce variable feeding regimes to better mimic wild-ish, I'll say wild-ish conditions, while in captivity, while maintaining very rigorous animal care guidelines, right? We still have to make sure we follow those. So the goal is to produce a more wild-like fish in terms of size, age of maturity, shape, body condition. So we have a number of various treatments. So on the bottom, there's various treatments, Monday to Friday, twice a day, Wednesday, Friday, twice a day, Monday, Wednesday, Friday, once a day, a 2-week random one that gets fed twice a day, the same random one that gets fed once a day. So we started this experimental feeding regime in September. So that's the gray boxes. And within 5 weeks, there was already noticeable changes in size, length, body condition, and even lipid content. And so this is very early. We're going to carry this all the way to maturity next fall. This is very exciting because I think what he's doing is he's innovating conservation rearing practices to produce more wild-like fish. And we know that a more wild-like fish is going to perform better in the wild once released, and that's the whole goal. Of course, this chair is not simply science and research, although that's a lot of fun, but it's also to train the next generation of science leadership in the challenges and opportunities that face conservation of protected areas like those in Parks Canada. So I think myself and UNB is a perfect conduit for this. We're able to attract students from across the country and around the world, and it's this diversity in backgrounds and approaches that has really led to solving problems of the future. So as a research chair, since I began this in 2019, I've had 16 students conduct research on Parks Canada sites. 3 of those have already found employment with other agencies. 8 of those students have moved on to advanced graduate degrees, either with myself or with other faculty in other universities. And 4 of those students have actually obtained full-time employment with Parks Canada. So I think this is a very exciting part, because as much as I want to keep all these wonderful students in my lab forever, that's not my job. My job is to train them, give them the opportunities, give them the experience, and let them move on to the world to help solve all the problems. Of course, there's those 3 pillars of the project, right? Salmon, aquatic restoration, aquatic ecosystem, and then that community engagement. And this social aspect, I think, it might be the most important of all the project, because if we don't get that word out, if we don't tell people what we are doing and present these results, especially to decision makers, then in large part, I think we fail. So I'm extremely fortunate to be associated with some amazing people at Parks Canada who do this extremely well. They drive a lot of the community outreach. I'm there to provide support and information where I can, help out as much as I can. But they do such an amazing job at this. I have to give them credit. There's so many different avenues, whether it's the Law Enforcement Coalition, the various salmon releases that are happening at different parks, salmon releases that are happening with our partners, in particular Fort Folly, whether it's magazine articles, TV programs, documentaries, working with communities. They do this exceedingly well. And so that is such a big part of this project. The last thing I just want to mention is, connecting people with salmon. We need people to connect. If people can't connect, I don't think they care. And so it's hard to make people connect with a species or an organism that you can't see and you can't touch. So programs like Swim with the Salmon, that takes people out into the river, teaches people about salmon, teaches them about conservation, monitoring methods, and in this case, snorkel surveys. But again, it connects people directly with salmon. Being able to get in there into their environment and see these fish, these endangered fish in their natural habitat, is extremely powerful. So that's a big one. Of course, there's salmon open houses in the various parks. There's all kinds of citizen science programming. All of this is designed to connect people with the parks, but in particular for these programs with salmon. I just need to thank my sincere, deep thanks to all the parks, all the field unit superintendents, the conservation and restoration program, the ResCon staff. Without them, this could not happen. I could not do the things that I do. I could not train the students that I train. They've all been very welcoming and put a tremendous amount of work into this multi-park project. And I think it's important that we all share in this collective accomplishment and just understand how truly impactful this project is. We've accomplished many things already, and I think these results stand on their own. And then lastly, I'm fortunate enough to sit in this chair position, but I am one small piece in a very, very large collective effort across Atlantic Canada, across these parks, to restore salmon, to restore aquatic ecosystems, and I'm very fortunate to be in this position. It is through such a collaborative, collective, mutually respectful effort that we are able to achieve big things. So with that, thank you all very much, and I'm happy to take any questions that you may have.
Well, Kurt, thank you very much for that presentation. It really touched many factors that we need to look at to improve the future for Atlantic salmon, from the improved applied research to how these results can be used as tools and models, and also the importance of, like you said, collaboration, stewardship, and just the next generation. So thank you very much for that. I'll be happy to moderate the questions and answers. Right now, there's two ways that you can ask a question. If you want to ask it yourself, you can raise your hand and I will permit you to do so by unmuting you. If you want to type in your question, I'll be happy to read it out loud so Kurt can answer it. So here, I'm just going to go look to see. I already see a hand risen, so I will go to that one first. So Aaron Allen, I will give you permission to unmute here. Second. So if you unmute yourself, you just got a request, you can go ahead.
Hi, Kurt. My name is Bill. Can you hear me now?
Yep.
Good. My name is Bill Haley. I'm calling from the Margaree Salmon Association. I'd like to commend you on the work that you're doing, and especially getting it out there, being transparent, and communicating with the public, with NGOs and other government departments. I do have a question. I see much of the work that is being done in this program revolves around fish being raised in captivity, whether it's in freshwater, onshore captivity, offshore saltwater captivity. Now, DFO have done away with all of the hatcheries on the east coast of Canada. Looking at the results that you're now achieving, is DFO giving any thought to actually having their own hatcheries in their own facilities for raising fish on the east coast of Canada, rather than going to Cook Aquaculture or going to Dalhousie University?
That's a great question. We do work with DFO at various aspects of all these projects. I don't currently know of any initiatives for them starting their own hatcheries or conservation farms like that's established in Dark Harbor. But I think it's an interesting idea, and I think our results are early, and I'm always guarded with our results. They're very promising, and we're very excited, whether it's in Fundy National Park, Cape Breton, or any of the parks really. But these are early, and we have to be a little bit guarded. But I think, I think that's a really interesting idea, and something I definitely bring up with them next time I talk to them.
I'll just mention that here in the Marguerite, we've had a hatchery probably since 1902, and we have probably the only salmon hatchery located on a salmon river, and we just happen to have the most healthy salmon population in the province. I don't think that it's just happenstance that happened. I think there's a direct relation between having a hatchery and having a healthy salmon population. So with that, I'll end my little spiel with thank you for the presentation.
Thanks.
Okay, we'll move on to the next question. Peter Sellings, I will send your request here, and you can go ahead. Peter? You're unmuted if you want to ask your question. Okay, I do have one that I'll move on to the next one. I don't know if there was a problem with the mic. We do have one written. The question is, what would be the best way to learn more about the citizen science programming that you mentioned?
Oh, that's a great question. So each park has their own kind of suite of citizen science programs. So I would encourage you to reach out to whichever park you're interested in going to. They will have a full list of each of those citizen science programs that you can participate in. So that would be the best way, is reach out to each park individually, whichever one you're going to attend.
Okay, thank you, Kurt. And there's another one that just came in. I'm interested in the salmon research open houses. Could the foundation share when these dates are posted for New Brunswick? Oh.
Yes, that's a great question. That would come from some of the Parks Canada communications people. So they help organize a lot of these things. So what I can do is I can reach out to them. They may or may not be on here, but I can reach out to them to see that when these open houses come up, that they get distributed through I know they're distributed through various channels already, but I'll see if they can distribute them through the Atlantic Salmon Conservation Foundation as well. That's a really good point. Thanks.
Yeah, and thank you for the question on our behalf as well. I see Peter has his hand risen again. I don't know if he wants to try to. There must be something going on with his mic, so I'm apologizing if it's not working for you here, Peter. I think he's on the phone, so he might need a pin. We got a couple more minutes for questions if there's anybody else that have a few questions. Okay, Eden Isbell, I will send you a request. And you are free to unmute. Aiden?
Great presentation. I actually didn't have a question. I guess it accidentally hit the raise hand button. That.
Okay. If anybody thinks of questions after the fact, please feel free. My email was attached with the GoToWebinar here today, so please feel free to send it to me, and I'll make sure that Kurt has it. And I should ask you, Kurt, but I'm sure you'll be happy to answer any questions if anybody has them. So yeah, all right. So
if you want to email me as well, I'm happy for people to do that as well.
All right then, so if anybody wants Kurt's contact information or already has it, please feel free to ask him any questions, and I can always help you with that. So once again, I just want to say thanks, Kurt, for today's presentation. I really appreciate you taking the time to join us and to do this. I know a lot of people appreciate these webinars and the information that's provided, and we surely appreciate yours today. So this is recorded today, so if anybody that did register wasn't able to attend, there will be a message sent out after the fact to remind folks that this is going to be on our YouTube page. I will put that this afternoon. So if you know someone else that you think might have this as an interest, please feel free to let them know. So once again, thanks. And I want to remind also that we do have more webinars to come. We do this up until March. Our next one's January 25th with David Rod, and he will be speaking on smoke to adult supplementation, a potential conservation tool in depressed Atlantic salmon populations. So please feel free to join us for that webinar session. So thank you to Kurt, and thank you to everybody that participated and joined us today, and looking forward to our next one. So thank you. See ya.
Thank you for having me again. I really appreciate it.
Okay.